Good morning, everyone. My name is Jill Slater, and I'm going to be presenting this today just to help you get ready to teach International A-Level Biology. This is the second module. I think many of you were probably at the first module on Monday. The agenda for this morning is we're going to have a quick review of the specification. We're going to look at assessment objectives and command words. We're going to have a look at some mark schemes and walk through them. You're going to do some marking yourself, and then we will have a look at examiner reports and the support that is available to you from Pearson. The aims and objectives are to understand the assessment objectives, understand the different types of questions, understand the mark schemes, and then to practice using these mark schemes on exemplar student work, and then to have a look at the support provided by Pearson. A little bit more information about Pearson, the world's leading learning company and the UK's largest awarding organisation. The international heritage stretches back over 150 years, and today we partner with schools, universities and employers worldwide, offering world-class, globally recognised qualifications to over 3.5 million students a year. And Pearson operates in 70 countries worldwide. We will start with a quick overview of the specification, starting with a look at the qualification. The IAL Biology is a modular qualification. There are a total of 6 externally assessed units or modules. Units 1, 2 and 3, which are the ones you can see at the moment, comprise the International AS level, the IAS. The content of these 3 units provides students with a grounding for A-level studies. Units 1 and 2 are each split into 2 main topics or themes, as you can see. Unit 3 is purely experimental skills acquired from the study of units 1 and 2. So as you are teaching units 1 and 2, you will be carrying out core practicals, additional practicals, enabling your students to develop the experimental skills required. If we look at units 4 and 5, these are the IA2 part of the qualification, and IAS and IA2 together give you the full international A-level qualification. So again, there are 2 units. The content of these units is focused around developing a good understanding of fundamental biological processes, and it's more demanding. You would expect to be covering these units in the second year of an A-level course. Again, units 4 and 5 are split into 2 main themes, and unit 6 is experimental skills acquired throughout the whole of the study of the A-level biology. We're going to look in more detail at the assessment objectives. Assessment objectives describe the type of learning that is being assessed. So when the exam papers are developed, they will, as the questions are written, they will be focused on a particular assessment objective. Assessment objective 1 is to develop knowledge and understanding of science. Questions matching this assessment objective will require students to demonstrate learning of biological knowledge and understanding that is detailed in the specification. AO2A is the application of the knowledge and the understanding of science in both familiar and unfamiliar contexts. Questions matching this assessment objective will require students to apply their biological knowledge and understanding. It will often involve the application of learning to a novel or unfamiliar context. AO2B is analysis and evaluation of scientific information to make judgments and reach conclusions. Questions matching this assessment objective will require students to use their biological knowledge and understanding to make sense of information provided in the examination. It will often involve the application of learning to a novel or unfamiliar context. AO3 is experimental skills in science, including analysis and evaluation of data and methods. All questions assessing practical skills will match this assessment objective, and they will all be in the Unit 3 exam. So, as well as listing the assessment objectives, this table gives you the percent of question or the percent of marks that will address each of the assessment objectives. So, AO1 in the IAS, it's between 36 and 39%. In the IO2, it's between 31 and 34, giving an overall percentage for the whole qualification of 34 to 37%. You will see that the percentage of AO1 is slightly higher in the IAS than in the IA2, whereas AO2 is slightly higher in the IA2 than it is in the IAS, reflecting the increase in complexity as you go into year 2 of the course. And if you look at AO2B, there is quite a big increase in the percentage of the IA2 course that is assessing AO2B. The practical skills are the same throughout the qualification. So the percentage here is converted into the assessment objective marks on each paper. The IAS papers 1 and 2, Unit 1 and 2 are out of 80 marks. 4 and 5 in IA2 are out of 90 marks. The practical papers are out of 50 marks in both cases. So this shows you the approximate number of marks allocated to each assessment objective. And again, you will notice that there's a little bit of AO1 on Unit 3, but it is predominant, and on Unit 6, it's predominantly the practical skills. And there's no practical skills on any of the other papers. Now we're going to have a look at the command words. Students should pay particular attention to the command words used in questions. These command words will identify the type of response required. For example, 2 common command words are describe and explain. Describe the cardiac cycle and explain the need for a cardiac cycle would require quite different responses. Sometimes the question can be explained, but the explanation is directly from the specification, so it will still be AO1. Appendix 7, the taxonomy, page 68 in the specification, lists the vocabulary or command words that will be used in questions. And your students should be very familiar with these because it will guide them towards the type of response that is needed. So if we have a look at the command words and assessment objectives, the command it identifies the common command words and the assessment objectives that are often used. However, The command words need to be interpreted in the context of the question or alongside the specification to identify the relevant assessment objectives. The assessment objectives should help you in your teaching and guiding the students to the type of answers they need. Students don't need to analyze the assessment objective, but what they do need to do when they're sitting an exam is identify the command word and take account of exactly what it's asking them to do. Some of the command words will almost always be assessing AO1, e.g. compare and contrast, complete, draw, state, name, give. That is demonstrating knowledge from the specification. Most, but not all, multiple choice questions will be AO1. The multiple choice questions can be used to assess AO2 as well. Some command words will be generally associated with AO2A, e.g. calculate and determine and show. Others, such as assess, comment, criticize, discuss, evaluate, are mainly AO2B. But you will see that there are some command words that, depending on the question, can be used to assess any of the assessment objectives. And AO3 will be any of the command words that are used. Describe, explain, suggest depends on the context of the question, whether it is AO1 or AO2. Now we're going to have a look at some assessment objectives, and we're going to map them to the question. The next one we're going to look at, I'm going to ask you to look at in more detail and to put it in the chat box. But for now, could you just jot down or think about what each of these assessment objectives, which assessment objectives each of these questions will be addressing? We're looking at the assessment objective mapping in that are taken from one of the SAMS papers. Obviously, on the exam paper, these exam questions will be in context and would quite often have data or information to accompany them. We'll go through these and then I will give you some to spend a bit more time on working out for yourselves. So, state what is meant by osmosis is assessment objective 1. It's a specification term, and the candidate should be able to recall the meaning of the term. State is almost always used for AO1 because in most cases it is taking it directly from the specification. Explain why oxygen molecules can pass directly through the cell membrane. Now, explain, depending on the context, can easily be either 1 or 2. But in this case, Candidates need to know the structure of the cell membrane, but they need to be able to apply this knowledge to explain why oxygen can pass through the membrane. So this one is assessment objective 2A. Calculate the volume of blood is also 2A because it is a calculation using data provided, and therefore it's considered to be an application of knowledge, and therefore AO2A. Calculations in general will be 2A, and 10% of the marks on any paper will be for calculations. 3C is explain the difference between the dissociation curves. Candidates will have been given the dissociation curve. They're asked to explain something about them. So this is considered analysis and evaluation of scientific information. So therefore it is AO2B. 5C, explain why each codon for the DNA genetic code must contain at least 3 bases. Although it's explained, from the specification, candidates are required to understand the nature of the genetic code. So in answering this question, candidates are demonstrating their understanding, which is in the specification. So the correct assessment objective is AO1. You've got 2 explain questions there. One is direct from the specification, so it's AO1. The other one is applying knowledge, so it's 2A. And then the final one, explain how this mutation results in a non-functioning CFTR protein. They've been given information about a mutation. They need to apply different specification points to provide an answer to this question. So the correct assessment objective is 2A. So I'm going to give you some more questions. I'd like you to have a look at these, and I will give you 5 minutes to put into the chat box the assessment objective for each one of these. we'll have a look at what the writer of this gave when the paper's written. Everything has to be balanced, so each question is allocated an assessment objective to ensure that there's the right spread of assessment objectives across the paper. So when the paper is written, assessment objectives are allocated, and this is what the writer allocated. I think you were all in agreement that state what is meant by the term phenotype is assessment objective 1. It's a specification term, and candidates should be able to recall the meaning of the term. State is almost always used for AO1, and terms that are stated on the specification, students are expected to be able to recall the meaning. Calculate how many times bigger. A calculation using data provided, therefore it's an application of knowledge, and therefore AO2A, which I think most of you put. A calculation is almost always 2A. Explain how the appearance of, I won't try and pronounce it, shows it is adapted to its habitat. Now, it's a bit more difficult when you haven't got the whole question, but this will be a diagram or a picture of this particular organism and some information about the habitat. So it is using biological understanding of adaptation and applying it to the information provided to them in the exam. So this makes it 2A. Draw 3 labelled features on this diagram that may be found in a prokaryotic cell. What assessment objective 1, because from the specification, candidates are required to know the structure of bacteria. So by drawing these structures, they're demonstrating their understanding, so the correct assessment objective is AO1. Explain the distribution of bacteria in the digestive system. Use the information in the diagram and the table, so they're obviously given some information to support your answer. Now, this was deemed to be to address both 2A and 2B. Depending on the mark scheme, when the grid is drawn up, there'll be a certain number of marks assigned to each of A and B. But one question can address more than one assessment objectives. They're applying their biological understanding and using this to interpret the information provided. So you've got applying knowledge, and then you've got interpreting the information provided. So it's both AO2A and AO2B. The final question, now you were a bit more varied in what you thought about this. Describe the role of the rough endoplasmic reticulum in the synthesis of hemoglobin. This is a straightforward application of biological knowledge. It is taken from the specification, so therefore the assessment objective is AO1. Now we're going to move on to look at language that can cause candidates problems. Many candidates produce answers that have lots of correct biology, but they gain relatively few marks because they're not answering the question asked. It's not incorrect, but it's not relevant to the question that they've been asked. And a significant reason for this is that candidates do not read the question carefully. As well as the command words, candidates need to consider the context of the question. Are they being asked to explain how something happens or why it happens? Are they being asked to calculate a percentage increase or a percentage decrease? Is the question being asked about a particular example, e.g. this protein or these enzymes? candidates really do lose marks by not reading the question properly. So if we look at this question, they're given information about 3 drugs that are used to treat blood clots. One of them binds to the active site of thrombin. So they're asked to explain how this drug reduces blood clotting. So it's one particular drug out of the 3 they're given, and they're told it binds to the active site of thrombin. Put into the chat box, it's a 2-mark question, it tells you that. What marks would you give these 2 responses? Right, this is a very good example of students reading the question and not reading the question. It is correct that platelets stick together when you get coagulation. However, that does not answer the question. They're specifically told that the anticoagulant binds to the active site of thrombin. So A is incorrect because they've not answered the question that they were asked, whereas B explains exactly how the anticoagulant that they are given the information on works by binding to fibrinogen so that fibrinogen cannot be converted to fibrin. A describes the action of how a different anticoagulant might reduce clotting, so it does not answer the question. And students do, these are taken from a genuine papers, students do lose marks because they don't read the question carefully. We're going to move on to look at what a mark scheme actually is. The mark scheme describes how a paper is to be marked, and it's quite precise, and it's produced at the same time as the paper. However, it is adjusted after the candidates have sat the exam to take account of the sort of responses candidates are producing. So it's never altered to accept something that either doesn't answer the question or is incorrect. But quite often students have put things that are correct and do answer the question, but weren't thought of when the original mark scheme was written. Or there might be adjustments made as to how much goes into each marking point. The mark scheme for the majority of questions in the IAL biology papers are point-based. This means that there are independent marking points for which a mark can be awarded. In general, for a points-based mark scheme, the number of marking points will be equal to or one more than the number of marks available. So for a question worth 4 marks, there will be 4 or possibly 5 marking points. And there's 1 mark awarded for each marking point. So if there's 5 marking points, but it's a 4-mark question, if you get 4 of those 5 marks, you'll get 4 marks. In each of the unit 1, 2, 4, and 5 examinations, there will also be 1 or 2 levels-based questions. The mark schemes for levels-based questions are different, and we will look at an example later. Think about how to use a mark scheme. This is an example of a points-based mark scheme. A mark scheme provides the answer for a question. Questions are marked by experts who interpret and apply the mark scheme to candidates' responses. There may be a number of options for a particular marking point, and sometimes guidance is provided as to what alternative wording is acceptable. In general, in a points-based mark scheme, each marking point is a standalone mark. This means the candidates don't have to gain the first mark in order to gain the second mark, and so on. This is a fairly typical mark scheme for a question. The first column identifies which question on the paper it is, the question number. The second column contains the answer. Each marking point is given as a separate bullet point with the mark in brackets after it. The additional guidance for each marking point is provided in the third column. So 2 types of additional guidance are provided: ignore and accept. These are self-explanatory, but the question was obviously based on fertilization and what happens after presumably what happens after the sperm cell reaches the egg. So the cortical granules fuse with the membrane and release enzymes. And when it says ignore chemicals, chemicals is not an alternative for enzyme. The word enzyme is required because that is a biological point. The zona pellucida hardens. Zona pellucida is in bold, which means that term has to be used, not anything else. But when it says hardens, thickens or becomes impermeable are acceptable alternatives to hardens. So the preferred answer is in the answer column, but the guidance is for acceptable alternatives. And again, the bottom one, accept no other sperm can enter. That's fairly self-explanatory. Now, We're going to look now at the meaning of the brackets on mark schemes because they have quite specific meanings. If they're in curly brackets, if we look at the first marking point, cortical granules stroke vesicles fuse with the egg cell surface membrane and release enzymes. To gain the mark, the student needs to address the full marking point. So in this case, they must mention cortical granules fusing with the membrane and release of enzymes to gain the mark. This is characteristic of mark schemes in the new IAL qualification. The curly brackets in the answer identify alternatives, one of which must be present. So the candidates must refer to either cortical granules or cortical vesicles. Smooth brackets indicate contextual information that may be omitted in the candidate's response, but which should not be contradicted. So in this case, it would be acceptable for candidates to describe granules fusing with a cell membrane and releasing enzymes. However, if they suggested granules fuse with the sperm cell membrane, that would be incorrect, the context is wrong, and the mark would not be awarded. A small number of 6-mark questions in Unit 1, 2, 4, and 5 papers use a level-based mark scheme. Level-based mark schemes have two components. First, they have the indicative content, an indication of the sort of credit-worthy comments candidates might make. Although indicative content can be a good indication of the quality of an answer, there may not be any correlation between the number of indicative content comments a candidate makes and the score awarded to the response. So the question was, explain what the solution of sugars should contain to preserve pineapples. Use the information in the table to support your answer. So they're given some information. They have to use both their knowledge and the information that they're given to construct their answer. On the paper, the mark will be starred, and it also is on the mark scheme. So this is an indication of the type of content that they might put in their answer. This is then divided or allocated to levels. So in order to maintain the sugar concentration, while preserving the sugar, you need correct conditions for osmosis to take place and correct conditions to maintain the sugar concentration. Two separate things going on, and you will be taking information from the table and information about osmosis to answer the question. So you've got two conditions. For level 1, you either need, to get the full level 1, you need a description of each condition or a description of one condition and an explanation of one condition. Each level is subdivided into 2 marks, giving you the total of 6. So to move on to level 2, you need an explanation of the conditions needed for both osmosis and maintaining the sugar concentration. And again, it's subdivided into 3 and 4 marks. And you need to fulfill each level before you can move on to the next level. So level 3 takes it further. So you need an indication that the concentration of each sugar needs to be the same as in the pineapple to maintain the sugar concentration, and that another solute needs to be included in order for osmosis to occur. So the sophistication of the response increases as you go from level 1 to level 3. They can answer they're not penalized for answering in bullet or point form. However, they need to make sure that they're actually giving it an explanation or a discussion rather than just listing facts. If they just list facts, they could penalise themselves. So, for instance, if they just stated that if they stated all the conditions, but they didn't give any explanation, then they are limited to level 2, no matter how well they describe the conditions. It's not, it doesn't mean that they don't have to write, they can write in bullet points, but it's easier to ensure that they've answered the question correctly if they write in prose. It's rare to find a 6-mark question that isn't level-based, but the level-based questions are clearly indicated by having a star by the question number on the exam paper. Now we're going to have a look at how the mark scheme is constructed. What you will see when you're looking at a mark scheme, past papers and mark schemes are available to you when you are signed up to teach Pearson Ed Excel. And it will help your students understand the assessment if not only do they have a view of past papers, but also the mark schemes that we used. So to consider how a mark scheme is applied, we will take a look at a paper and a mark scheme. And you're going to be doing some marking yourselves in a few minutes. So there is general guidance common to all the mark schemes for the information for examiners when they are marking the papers. The guidance is designed to encourage positive marking that rewards candidates for what they know. They will be marked on correct information that matches the mark points in the mark scheme. If they have added additional information that is irrelevant but is not incorrect and doesn't contradict anything that they've put, then that is ignored. They were looking for positive marks. And team leaders should be consulted if there's any doubt. If work is crossed out, it can be marked, but not if there's an alternative response. So if a candidate has just crossed out a question and then carried on and not done anything else with that question, it will be marked if it can be read. If they've crossed it out and then put an alternative answer underneath, it will be assumed that the alternative answer is the one that they want marked, and that is what will be marked. Right, now you're going to do some marking now. So if you look in your delegate booklet, you will find that there are 4 responses to this question. This is the mark. The question was, describe the conclusion that can be made about the risk factors for CVD. Use the information in the graph to support your answer. This is the mark scheme. It's a 2-mark question. You've got 4 responses in your delegate booklet. So if you could now mark those 4 responses and put the marks you would give them into the chat box. And we'll give you 5 minutes. You probably won't quite need 5 minutes for this question. We'll give you a few minutes to answer this when we've got plenty of responses in the chat box, then we'll have a look at the marks that were given. Note that increases appears in the mark scheme for both marking points. They need to refer to smoking and age increasing the risk, not just that it causes it gives the risk, but you have to have the increase. Right, when you've had a look at them, put me some marks into the chat box and then we will talk about it. Have a few more in the chat box and then we can talk about them. I've got a couple of comments in the chat box about people not understanding about level-based marking. We are going to look at some examples of level-based questions as we go through marking responses. So hopefully when we've done that and explained it, it will make it clearer to you. But if not, we'll come back to it. I'm not yet getting any responses to the marks for these 4 questions. If a question is marked with an asterisk, it means that that is a level-based question and will be marked levels will be given, and then a decision made within the level which of the marks to award. But we have got, if you look in your pack, the final questions that we're marking are level-based questions. So hopefully that will help explain it to you. So if you look at the questions in your pack, you should have a resources button that will make these packs available to you if for any reason you haven't got them or haven't printed them off. Right, we will move on and have a look at the marks that were awarded for them. Right, so response A was given 2 marks because it clearly states that the relative risk of developing CVD increases as age increases, and the risk of CVD of smokers is higher than that of non-smokers. There's a comparison there. The same with response B, the relative risk of CVD increases as age increases, and there's a relative higher risk value than non-smokers. Response C has only 1 mark because smokers have a higher risk of developing CVD than non-smokers. The second statement just picks out 2 things from the graph, but it doesn't. It says 45 and above have the biggest effect on smokers. It doesn't clearly state that age increases the risk of CVD. The same with response D. Smoking is a risk factor for all ages, no comparison. The group for smokers had a higher relative risk than of developing CVD. Clear comparison. In the past, the previous specification, marks were quite often awarded for taking information from the graph, just quoting particular points on the graph. But with the new specification, that is no longer the case. Now, if you could look at the next question, it's a Punnett square, a genetic diagram. Rabbits can have brown fur or white fur. A heterozygous rabbit will have brown fur. So that tells you the brown is dominant. Straightforward genetic diagram to show genotypes and phenotypes of the baby rabbits produced if 2 heterozygous rabbits were bred together. It says draw a genetic diagram. So if you don't, you can only get mark 0.3. It specifically says draw a genetic diagram so that you can demonstrate that you know the genotype of the parents or the alleles in the gametes, and then the genotypes of the offspring. So we'd like candidates to follow the convention of the capital for the dominant trait and lowercase for recessive trait, but if they use a different notation, as long as it's makes sense and is applied with appropriate logic, that is acceptable. The additional guidance shows 1 version of a diagram that would gain all 3 marks. But a Punnett square is fine. There are other alternatives. So if you could mark the 4 responses that you have got and put them into the chat box. few more responses into the chat box would be good. Is there anybody having difficulty seeing these resources? I hope not. CE means carry error forward. So if you've made a mistake early on, then that can be carried, that can be used. They would lose the mistake mark for where they made the error, but it doesn't mean they lose all the subsequent marks. Quite a bit of variation in the marks you awarded. Right, the first one was awarded 3 marks. It clearly gives the parental genotypes, offspring genotypes. They can be inferred from a correctly drawn diagram, and then it gives the percentage of each type of each colour fur. Again, all 3 marks can be awarded. They went off on the wrong tack, so they didn't get any marks at all for C. The final one, 3 marks again can be awarded. This type of diagram is just as acceptable as a Punnett square. They give a key, so they make it clear, although they've not quite followed the normal convention, they give a key which makes it quite clear, and they label genotypes, they label phenotypes, so they can have 3 marks. This goes on to give a calculation for a number of heterozygous pairs of rabbits were bred together, number of rabbits produced. You've already worked out the percentages in the previous question. So calculate the expected number of homozygous brown rabbits, heterozygous and white. And there are 3 answer lines asking for 3 different responses. And those are the numbers. If they have put 1 to 2 to 1, they haven't done the calculation. They haven't calculated the actual numbers of each one, but they can have 1 mark for showing that they know the ratio. So again, if you could mark, there's 2 responses. If you could mark these quickly, put the answer in the chat box. Yeah, this is fairly straightforward. The first response gets 3 marks. You've got all 3 answers on the answer line correct. The second one it's only written the right answer on the bottom line. There's no consequential error or error carried forward when lines are provided for each mark. If there's just one answer line and one answer, then there might be marks in the working out. But for this one, where you can't tell from the working out which one they would be referring to, so you just look at the answers on the answer line. Now we're going to look at a slightly more complex question. It's an explain question. It asks about, it's using information they've learned about red-green colour blindness to apply it to haemophilia A. Candidates often struggle to express complex biological ideas. The additional guidance column gives an indication of what is considered reasonable to accept in candidates' responses. Note the use of curly and smooth brackets to identify required and contextual parts to an answer. Remember, a term or idea inside curly brackets is required, whereas something inside smooth brackets can be omitted as long as the context is not incorrect. There are 3 marking points for 3 marks. So they have to have either gene or allele for mark point 1, but defective is in brackets, so that's just a context. Either gene or allele is accepted for mark point 1, but for the second mark point, it's got to be allele. And again, allele is in bold. It is a correct term which has to be used. Right, so if you mark the questions that you've got in your booklet, put them into the chat box again. These take a bit more reading, but if you put what your responses you would give them in the chat box, then we will have a look at them. We could have a few more ideas of what marks you would like to give them, and then we'll move on and have a look at what marks were awarded. We're getting a bit of variation in the marks awarded. We will have a look at which marks were awarded, and I'll explain why. It is the case that before examiners start marking, they will have looked at examples and had discussions about examples. So they'll have a lot more idea as to what is going to be accepted and what is not going to be accepted. They'll have more consideration of it before they start than I'm giving you this morning, and looked at more examples of what students have written. So the first one was awarded 3 marks. Candidates often write it, and it's obvious from the context of the question, it is recessive. The question is about haemophilia, so that mark can be awarded. There was nothing relevant to the mark scheme in response B, and quite a lot of it was actually incorrect. C was given 3 marks. The 3rd mark was borderline, higher chance of having a healthier X, being just about okay for the additional guidance. Many candidates struggle to express marking point 3 clearly. Responses C and D are examples where marking point 3 was awarded, but it is easy to see why some examiners may have been reluctant to award the mark. It would have been easier to award if candidates had expressed the idea more clearly. Marking is consistent as far as is humanly possible. The main thing is to have everybody marking to the same standard, but you will always get responses that don't nearly but don't quite match what's on the mark scheme, and a judgment has to be made. But if you encourage your students to write as clearly as possible and use as much correct terminology, then they will make sure that they get the marks that they deserve. We're going to briefly consider one of the multiple choice questions, just so that you see how they are put together. 10% of the marks in each of papers 1, 2, 4, and 5 will be for multiple choice questions. They all follow a standard format of a question stem followed by 4 options, only one of which is the correct answer. There's never more than one correct answer. The mark scheme gives the correct answer and a brief explanation as to why the other options are not correct. That is to enable, when the paper is being written, to ensure that it's not possible for any of the other answers to be correct. We're now going to look at a compare and contrast question. Amylose and amylopectin are components of starch, both on the specification, both testing students' knowledge. Compare and contrast the structure of amylose with the structure of amylopectin. In order to gain full marks, candidates need to give both similarities and differences. The mark scheme has 2 similarities and 1 difference for 3 available marks. You will see quite a lot of mark schemes with 2 similarities and 2 differences, but never more than that, so that they've got to give one similarity and 1 difference in order to get full marks. The additional guidance is to not piece together, so they can't just list all they know about amylose and then all they know about amylopectin. It has to be a comparison. And then there's other things that are allow or accept. And then a comment at the end. So if you mark the ones you've got on your, you've got 4 in your pack. If you could mark those and put the marks you get into the chat box, and then we'll have a look at them. We'd have a few more responses and then we'll move on, because you've got quite a few questions to mark. NB says, please note, if they've written amylose has 1,4 glycosidic bonds and amylopectin has 1,4 and 1,6 glycosidic bonds, that is sufficient for mark point 2 and mark point 3. They may well, if they write it correctly, they can answer this question in a very, they don't need an awful lot of sentences to write it. We'll have a look at the marks that were awarded. So the first response gained mark two, mark point 2 and mark point 3. Clearly express the idea of getting similarities and differences in bonding, but the mark point 1 requires alpha glucose, and there's no reference to alpha glucose, so not mark point 1. Response B got the bonding marks, mark point 2 and 3, but again, it missed the alpha in front of glucose, and so did not get mark point 1. C gained all 3 marks. Although, if it's read by itself, the first sentence is not correct. Amylose and amylopectin are polymers of alpha glucose, not alpha glucose monomers. But in the context of the question and what they went on to say, it seemed fair to interpret the sentence as amylose and amylopectin are made from alpha glucose monomers and allow the candidate to have mark point 1. This is an example of a situation where an examiner must make a decision about the way a candidate has expressed themselves. And again, different examiners might come to a different decision. D gained mark point 2 and mark point 3. Glucose molecules, no alpha, joined by 1,4-glycosidic bonds. Amylopectin, you've got whereas, or meanwhile, so you're comparing them and you've got the different bonding in amylopectin. Right, if we move on to the next question. There's obviously some context to this. It's an explain question. And to gain full marks, the student has to produce an answer that's got an element of explanation. So explain how uptake of substances would be affected if the amoeba increased its number of pseudopodia, tells you what increasing the number of pseudopodia will do, and then therefore uptake or diffusion would be faster. You've got to have the idea that it's faster or the rate increases. Uptake itself is not greater. It's not going to uptake more ultimately, or it doesn't increase. You need something to do with faster or rate, and ignoring any any reference to active transport because it's not. So if you could have a look at responses A, B, C, and D. And again, put me some marks in the chat box. Students will get the mark if they write it as points, as long as their points are correct, yes. We'll have a look at the marks that were given. So A, B, and C gain 1 mark each. Mark 0.1 for an increase in surface area. Statements like uptake of substances would increase or uptake would be more were not allowed for mark 0.2. It's got to be an increased rate rather than, I don't know why this is, it was given 2 marks because it's quite clearly only mark 0.1. Surface area increases. In response B, the use of it was accepted in place of uptake, but it's got to be an increased rate. And D gained both mark 0.1 and 2. Increased surface area to volume ratio was accepted in place of just increased surface area. It's the increase in surface area that's the relevant thing, but surface area to volume is accepted. There are instances where you have to have surface area to volume. Right, we're now going to come on to the levels-based question we're going to have a look at, and I wanted to make sure we had time to discuss this in the level of detail needed. 6B, and then you can see the star between the B in brackets and the 2 in brackets. So that indicates it's a level-based question. Explain the possible effects of these 3 types of mutation on the amino acid sequence coded for by this length of DNA. Use the information in the table to support your answer. So they have got a table which gives them 3 types of mutations, and it gives them a particular length of DNA. It tells them to use the information in the table. In order to gain full marks, they've got to refer back to this specific information. They've got to apply their knowledge to an unfamiliar situation. So if we have a look at the indicative content, the 3 types of mutation that they're given information on are substitution, deletion, and insertion. So for each one, the indicative content gives you a little bit about generic information about each type of substitution. It then goes on to say what the specific substitution that's given in the table has, what effect that has on that amino acid. So you've got a combination of generic information, but you've also got quite a lot that comes from the table, and they need to refer to the information on the table to get the full marks. This gives an idea of some aspects that they might want to consider. Then we get on to the levels. So for level 1, 1 correct statement about mutation. So 1 correct statement about what mutation, a generic statement about what effect mutations have on the sequence of the DNA. The second one is 1 aspect commented on. So if we go back to the aspects, substitutions change the amino acids, etc. one aspect commented on with a corresponding illustration from the information they've been given, or two or more aspects commented on, but no link to the information they've been given. And then this becomes more complex. For 3 marks, 2 aspects plus illustrations, or 3 or more aspects, but only 1 or 2 illustrations. So when you get into level 2, you've got to be using some of the information that they've been given, as well as their generic information, to achieve level 2 at all. And for full marks at level 2, we're expecting 3 aspects commented on with corresponding illustrations. And then when we get to level 3, it's 4 aspects and 5 aspects with corresponding illustrations. Give it a go on the examples you've got. You've got 3 examples. If you could put your the mark you would award into the chat box. If you've got any specific questions, put them in the chat box. And then we will talk about it. But I would like you to have a look at it and have a go before I tell you what was given. Right, we will have a look at the marks that we were awarded. From experience as a marker, these questions are often not particularly well answered, although as the specification becomes more established, the answers are improving. So response 1 or response A was limited to level 1 because it doesn't really address the question. On line 7, the candidate seems to be expressing the idea that substitution mutations may have no effect. So they've got the idea possibly that substitutions don't have an effect, although the attempted illustration was incorrect. Now, unfortunately, because you've not got all the information in front of you, you would not have been able to tell that, but it was considered to be a level 1 response and was awarded 1 mark. They've spent too much time explaining the effect on the protein that was produced and not enough on answering the question. Response B is a much better attempt at answering the question. They've included addressed several aspects, including substitution changing an amino acid and insertions and deletions changing sequences, as well as the idea of creating a new stop codon. So they've got several generic things about mutations in there. Unfortunately, only one of the aspects was illustrated, substitution resulting in aspartic acid instead of glutamic acid. That meant the response was limited to a level 2 response. 3 or more aspects commented on. So you've got the 3 aspects, but only 1 illustration. So the response was given 3 marks. Response C, this response, the candidate had addressed a number of aspects, including substitution sometimes having no effect, while insertion and deletion change the sequence. However, the candidate has not illustrated any of the aspects addressed. So this limits the response to level 1 and gets 2 marks. If you look back at these, are taken from the SAMS paper, so you will be able to have a look at those with your students, and past papers will also be coming available, or have started to become available to show your students. For the levels, you have to use, to get the higher marks, you've got to use all the information that's provided and apply it to your own knowledge. So just writing what you know about different types of mutation will limit you if you don't apply it to the information you've been given on the exam paper, which is why the final one was limited to level 1. And yes, to gain full marks, they do need to address all the aspects, but the key thing is using their knowledge but applying it to the unfamiliar situation of the information that they've been given. They must bring in the information that they've been given, and where they're given 2 lots of information, they've got to bring in both parts of the information they're given in order to gain full marks. This is how these level-based questions are constructed. I hope that's made it clearer to you. The more you look at with your students, the more familiar you as well as your students will become with them. And we're also going to finish this session by looking at examples of examiners' reports, which will also help you understand how these questions are marked. We've got two more questions to look at quite quickly because I do want us to have some time to look at the examiner's report. So if we move on to, this is a determine question. It's a new command word in this qualification, and it must have an element which is quantitative. It must have some calculation. So the given information, there's two marking points. The first one, they've got to calculate the difference between the systolic and diastolic pressure, and then link that to the information they're given to say whether or not the person is healthy. So you have got a number of responses. You've got 4 responses, quite straightforward to mark. If you could have a look at them and put the marks into the chat box fairly quickly so that we've got time to finish everything. We will move on and have a look at this because it is quite straightforward. Right, so for the first one, it got both marks. They calculated the pulse pressure and 25% of the systolic pressure and made the correct interpretation. So it gets both marks. B and C didn't get any marks. They had incorrect pulse pressure interpretation, incorrect calculation, and incorrect interpretation. In B, the pulse pressure was incorrectly calculated, but there's no indication of the 25% of systolic pressure, and the statement about being unhealthy is incorrect. In terms of what they calculated, it's incorrect to say that they're unhealthy. And again, with C, the pulse pressure 93% is not correct, and the consequential interpretation is also incorrect. And D gained 1 mark for the correct calculation of pulse pressure. Now, the command word in this question, the next one, is suggest. Candidates are expected to use their biological knowledge in an unfamiliar or novel context. The suggest command word will be used for no more than 10% of the available marks in the paper. So they're given some information about an enzyme. They should know about the urea cycle. They're given some information about a deficiency. Suggest how a person can be shown to have this disorder. So it's actually about the tests that can be done to show that a person has this disorder using the information that they've given. Ignore the additional guidance says ignore where the molecules might be found. Because it's unfamiliar context, they should not be penalised for suggesting measuring a molecule in an inappropriate location. So if you're measuring it in the blood, liver, or kidney, all acceptable. Or if you're suggesting it without a specific location. So a couple of minutes at most to have a look at these and put the marks that you give them in the chat box. There are 4 responses, so let's have some marks in. These are picking up slightly more marks than you seem to be giving them. 3 of them have commented on a biochemical substance that will change because of the deficiency. So the first one, we've got carbon malphosphate cannot be produced. Citrulline will not be produced. High ammonia concentration, all acceptable because these are all molecules that are part of the urea cycle. There's an example of not really reading the question properly, because not referring to how it can be shown that they've got this condition. Because it suggests it's something that might happen or might be able to be done. So they don't need to know, they just need to know how tests can be carried out for a biochemical disorder. And they also need information about the urea cycle. So those are the marks that were awarded. Now, at the end of the exam series, the principal examiner on each paper produces an examiner's report. It's a very useful document. It considers the performance of the paper for the cohort as a whole. It draws attention to areas in which candidates did well and those in which they did not. It includes consideration of the marking points that candidates gained or didn't gain, and candidates' candidates' responses are used to illustrate why particular marks were or were not awarded. So this is an example. The examiner's report has a comment on each question. The question in this case was, explain the importance of each of these ions to the plant. It says nearly all candidates could explain the importance of nitrate ions. Fewer could fully explain the importance of calcium and magnesium. Many stated what they need, that they made calcium pectate or made chlorophyll, but they didn't expand the explanations and say why that was important. And a small minority described what would occur to a plant deficient of these inorganic ions, which may be biologically correct, but is not answering the question. You may find it helpful to share these reports with your students so that they can see what sort of things the examiners are looking for. There's also an exemplar response in the examiner's report with comments about what marks were awarded and what marks were not awarded. So it scored 1 mark for the importance of nitrate ions, doesn't say why chlorophyll is needed, and no reference to calcium ions being needed to form calcium pectate, so that mark would not be awarded. This is an example of feedback on a level-based question. It says exactly why their levels were limited. Now you have got access, you will have access to this presentation afterwards, so you can go back and have a look in more detail. Occasionally you get, again, it's reading the question carefully. It's obviously about drug testing. A minority of candidates gave a description of the antimicrobial core practical methodology. So some students will go straight into describing a practical which is not relevant to the question they're being asked. Sometimes if it is a practical question, they will describe the wrong practical. So just imprint into them to read the question carefully and really think about what it is asking. an example of a question, this is an example of a good answer. It's a level 3 response. They've just missed out testing in vitro or statistical analysis. These are questions from no, they're not Sam's papers, because otherwise there wouldn't be an examiner's report. We're just going to finish off with a quick look at the support that is available from Pearson. They produce, and when I've done this, I'll have a look at what's in the chat boxes, so I will answer your questions at the end. They produce free resources and support for planning, teaching and learning, for exam preparation and assessment, and for results support. And it lists the resources they produce and which area they will be useful for. They're available on the website, and the web page has 4 tabs where you will find specification and sample assessment materials, exam materials, and in the exam materials there are past papers, mark schemes and examiners' reports, forms and administration, and then teaching and learning materials, more exemplars, lesson plans, course planner, and much more. After the exams and in preparation for exams, Results Plus is a very useful tool, allowing teachers to analyse their students and their own teaching performance. So you can see in-depth data on the performance of a candidate, a particular candidate, a class, or a school in reference to the cohort. So this is useful to see which questions and spec points students have performed well in and poorly in, and use this around revision and future classroom teaching. And it can also be used for mock exam results. If you give your students a mock exam, you can feed their results into the system, which will help guide them in their future revision. It's a free online service to Pearson Edexcel centres. It was developed in 2007 when ePen was first used. Students take their exam in a traditional based way. They are then sent to a central processing centre and put through a series of machines. Electronic copies are then sent electronically to examiners. So that can all be consolidated and used to produce performance reports, which can be shared with teachers. Exam Wizard is another very useful tool to produce exercises and past papers for students to use for study or in classroom tests. It uses questions from past papers, so the bank of potential questions increases with each series, and you can use it to produce bespoke questions or question papers, depending on what you're selecting to test. Access to scripts is self-service, online, free, and instant from results days onwards. Cuts down on speculative EARs, as schools have time and information needed to make an informed decision. It allows you to see all of your students' marked exam papers free of charge. It's accessible through Edexcel Online. The scripts don't have any examiner annotation on them, but they do have the marks awarded for the question. You can use the Access to Script service along with Results Plus to get a detailed breakdown of your students' performance in Edexcel exams, which will help you identify topics and skills where your students could benefit from further learning. Pearson Publishing. Pearson produces textbooks and various other resources. It supports a modular approach in line with the latest specifications, which are recognised by universities worldwide and fully comparable to UK reformed A-levels. Provides exam practice, appropriate international content, It's got embedded transferable skills, and it's reviewed by a language specialist to ensure that the book is clear and accessible. And any further questions, you have a dedicated subject advisor. I think you'd have to look into it, and if you go onto the web page and look at Results Plus, it will give you more information. I'm not quite sure whether you scan the mock papers or whether you just put the marks in that you've awarded, but it's certainly worth looking into. I think that brings us, or that does bring us to the end of the presentation. I'm sorry it got a little bit rushed at the end, but I hope that has helped you understand more about marking and how to prepare your students for exams. And if I close that down for a second, you can contact Tim, your subject advisor, with any further questions that you've got, either on the telephone, either by phoning him or by emailing him. Hope you enjoy teaching A-level biology.